My name is Theo Knijnenburg. I work at the Allen Institute for Cell Science, and today I will talk about a deep generative model for single-cell organization. I want to thank Luciano, Quinn, and other organizers for the opportunity to present, and also Luciano for your kind introduction, bringing me back all the way to my yeast days in Delft. Okay. At the Allen Institute for Cell Science, we want to understand the cell by modeling the cell. And in first instance, we do this by looking at the cell. Of course, acknowledging that cellular organization is tightly coupled to cellular function. We are interested in basic research questions around the principles of organization and how to sensibly model cell states and their transitions. This basic science component naturally flows into applied research questions regarding differentiation and disease. So we take an integrative approach, considering all the main parts of the cell at the level of organelles, which really requires 3D live cell imaging followed by quantitative methodology. Our model system is the human inducible pluripotent stem cell in its undifferentiated form. We want to visualize the morphologies, the positions, and dynamics of all the key structures in the cell. And we want to do this in as unperturbed cells as possible. So we use endogenous GFP tagging to create very carefully QC'd cell lines that we can then image. Imaging is really at the core of our institute, and we have developed sophisticated, reproducible, and scalable microscopy pipelines. As we typically tag only one structure per cell line, we want to integrate image data across experiments to get a picture, or perhaps better, a model of cell organization. But we actually go beyond just imaging data and also measure transcriptomics data. This is especially exciting when we differentiate these stem cells to highly organized cardiomyocytes, as we have the opportunity to follow and to integrate the informational layer of the transcriptome with the functional layer of subcellular structures. Everything we do is quantitative in order to enable analysis and modeling with the explicit goal of empowering and sharing with the research community. The one take-home message today is allencell.org. This website gives you an excellent idea of what it is that we are doing and what we have available in terms of data and tools. Now, I wanted to give you a second take-home message, which was a link and a QR code to the bioRxiv submission of the paper that I will talk about today, but not yet. If you are interested in the paper, it will be available via allencell.org soon, and I would also be happy to point you to it. Would it not be great if we can see with a microscope the cell and all its internal structures as the cell goes through cell division or cell differentiation? Experimental limitations and cell health or toxicity, if you will, prevent seeing this, at least at this point in time. And this is where machine learning can play an important role in filling in these experimental gaps. So we have dozens of cell lines for which a single structure is tagged, but how do we integrate this information? And this is one of the main questions that we are trying to answer in building an integrated cell. We are studying 4 different approaches to integrate information across structures. Very briefly, there is the idea of superimposition. We have many cells, many cells. And so also we have many cells that look alike. And we can superimpose cells with different organelles imaged in order to get multi-organelle composite images. We are also looking into rule-based approaches. From relationships between organelles that we can infer from our large data sets, can we now derive rules that we can sensibly use in order to pack organelles or subcellular structures inside of cells and create useful in silico models? And then there are 2 deep learning approaches that we have developed and that we are using in the Institute. One is label-free. We call it label-free. It's a typical image-to-image based approach where we predict organelle location and shape based on the bright field channel using a 3D UNet architecture. And the second is a generative model using a beta variational autoencoder. And it is this model that I will talk about today. I will start with giving a sort of quick summary of the points that I'm going to talk about. Data. So we have more than 40,000 three-dimensional images of cells where we have obtained 3 intensity channels: the nucleus, the cell membrane, and an organelle or subcellular structure. We are, as a computational model, using a beta variational autoencoder architecture in order to model the cell and the nuclear shape, but also to model the organelle shape and location, which is conditional to the cell and nuclear shape. In our paper and in this presentation, I will present a whole bunch of applications which we implemented in order to understand what happens when we push these 3-dimensional cell pictures through the model. How can we interpret the latent space? What happens when we turn the knob on beta, which gives us a trade-off between the sparsity and the reconstruction capabilities of the model? We can, of course, generate pictures of organelles now with our model. We did a study towards the statistical coupling of organelles to the nucleus and to the cell membrane. And we did a small application of out-of-sample data, drug-treated cells that we ran through our model. We are very interested how we can make this work into an actual scientific tool that can contribute to the larger projects that we have ongoing at the Institute. At this point in time, we are employing the beta variational autoencoder to describe the shape of nuclei during colony formation, so using time-lapse data. I was not planning to talk about this, but I'm actually quite excited to do so a little bit after Virginie's awesome presentation. What we are doing is we have 3-dimensional black and white binary images of nuclei. We can run these 3-dimensional images through the autoencoder. At the same time, what we are doing is we are trying to parameterize these shapes that are sort of underlying these binary images. We mesh the nucleus and then apply a spherical harmonic decomposition, which is a sort of 3D version of a Fourier transform. And then we actually run the spherical harmonic coefficients through an autoencoder as well, and can then regenerate actual shapes. So very exciting work, and I think very close in a way to what Virginie was describing. Paper is almost there. I hope to share it with you soon. Okay, let's dive into the data. In our typical imaging assay for these undifferentiated stem cells, we put a dye on the DNA and we put a dye on the plasma membrane in order to see the nucleus and to see the cell membrane, right? And we do this alongside a GFP tagged structure. So the DNA is here seen in cyan color, the plasma membrane in magenta, and then in white we see the mitochondria for this particular Z-stack video. In the 3D reconstruction on the left, we get an idea of what it looks like when we see these cells grow in a monolayer. Okay, so we have thousands of these 3-dimensional images of single cells. The cell images are registered to fit within a uniform sized box, which is 128 in X, 96 in Y, and 64 in Z. And so once again, we measure 3 channels. We measure the cell membrane dye, is here indicated in the magenta color. Again, the DNA dye in the cyan color, just pointing out a few of them. And then in yellow, in this particular case, we have a, if you will, rotating cast of 24 different GFP tagged organelles. And you see 3 different examples here: mitochondria, nuclear envelope, and tight junctions. So Our computational model consists of two parts. The first one, we call it the reference model. So this is a typical autoencoder model that consists of an encoder, right, which encodes the about 1 million voxels that we have in such an image into a latent representation that is much smaller number of dimensions, giving us ZR, which is a generative model. And then we have a decoder that takes this latent representation and returns a reconstruction. This reference model takes as input two channel images, or two channels, right? Namely the cell die, if you will, and the nuclear die. And we have this available for all our cells. We call these the reference channels. Okay, the second part is the target model, and this is where we have the organelle structure channel coming in, but in a conditional way. We are trying to encode the GFP intensity of the target structure, which can be one of the 24 organelles, conditional to the reference channels. So now that we have trained these models, one direct application is to visualize cellular structures by generating images sampled from these models. Here we are looking at 3 out of the more than 40,000 cells on which we trained, and we are looking only at the cell and the nucleus. And perhaps to briefly explain how we are looking at this, we are using two-dimensional layouts to show these three-dimensional cells. Specifically, we are looking at 3 maximum intensity projections. One is from the top. This is the XY plane. And then we have 2 from the side, XZ and YZ. Okay, so now we can generate cells and their nuclei by sampling from the latent space. And so here we have 7 such examples. And I think it looks pretty nice. The pictures would have been even prettier if we used a GAN, a generative adversarial network, to try and make real pictures indistinguishable from generated ones. The previous instantiations of our model actually had a GAN component to them, but we found that models without a GAN improved numerical stability of the optimization. It was easier to get the model to train and to converge. And it was also more straightforward to interpret the latent space without having a GAN. Okay, now looking at the conditional model of the target structures. So the construction of the model enables us to impute and visualize multiple cellular structures in the same cell geometry simultaneously. Whereas the data, as you know, is trained on only images that contain one structure at a time. Okay, so here we are looking at the same 3 cells, now with a measured GFP channel in there, right? So the first cell was a cell that was tagged for the mitochondria. In this composite image, you see it here in yellow, and in the left-hand side, you see it separately. Here we have an image of a cell which is tagged for the nuclear envelope, and the third one is tagged for tight junctions. The tight junctions are usually at the apical surface of the cell. These are apical components of cell-to-cell junctional complexes. And if you can see it when you look over here, the laser pointer now is sort of at the bottom of the cell of this YZ projection, and now I'm moving up. So here I'm at the top of the cell, and that is where you see the yellow of the tight junction. I hope that is visible. So now we can generate these organelle GFP intensities for these 3 organelles in these actual cells or in generated cells. Here we see the intensity distributions for cells sampled from the latent space. And so we have 3 each for the various targets. Overall, we find that in distribution, the generated structures vary in localization as one would expect, which provides strong evidence that the network is successfully learning appropriate rules or patterns governing structure-specific localization. So specifically, the mitochondria are distributed throughout the cytoplasm, but are never found inside of the nucleus, although it's difficult to see from these projections. The nuclear envelope forms a closed shell around the DNA, and the tight junctions are at the apical surface of the cell. And you can see that a little bit here, where you see once again that the yellow is in the top of this projection. I am not sure to what extent you are surprised by these results, or whether the power of deep learning on microscopy images is already something that you take for granted. I think it is quite wondrous. Also, very much the work that Gigi just showed us. We are just throwing pixels or voxels, if you will, into this thing and not telling it anything else, and it produces sensible predictions of organelle location and shape. Okay, so let's talk about the interpretation of the latent space. So we go from 1 million voxels to a 512 dimensional latent space. So this means that first a picture is represented by 1 million numbers, and now it is represented by 512 numbers. Here we are looking at the two most informative latent space dimensions, mu71 and mu419. And each cell is plotted as a point in this two-dimensional space. We see if we color these points by their mitotic state, the cells in various mitotic stages nicely stratify based on these two dimensions. It's perhaps important to mention that about 5% of our cells are undergoing mitosis as we take a picture of them. Another example, here on the right-hand side, we are now coloring by cell height and clearly see the relationship between the latent space dimension mu419 and height. And so by correlating the latent space dimensions with various metrics and observations, we can get a biological interpretation of these dimensions. A beautiful feature of these variational autoencoders is their generative capability. Pick any point in a latent space and generate cell visualizations from that point. We often create what we call latent space walks, where we can fix all the latent space dimensions but one, and then sample from various points, in this case from -2 standard deviations to +2 standard deviations, along that dimension in order to better understand the variation. So for example, when we are looking at this latent space dimension mu419, we indeed see that the height of the cell increases when we walk along that dimension. Remember, once again, this is the YZ projection, and so we see that Z is increasing as we walk along this dimension. So mathematically, the goal of the beta variational autoencoder is to optimize the evidence lower bound, which consists of two parts. One is the ability to reconstruct, and the other is the complexity of the model. And you tune this by tuning this beta parameter. We see that only about 50 dimensions out of the 512 actually contain information about the cell pictures. And this is due to this model complexity term that the other dimensions are closer to the uninformative normal distribution. A final interesting point that I want to make about the latent space is that the dimensions are statistically independent of each other. This is not directly actually by construction, but this falls out of the math and also out of the actual experiments as we have done them. So whereas the voxels in the original image are highly correlated in the sense that neighboring voxels tend to have the same values, these dimensions are disentangled, and this very much facilitates their biological interpretation. So we can tune beta, and we did this for two-dimensional models. Right, so each point that you see in this particular plot is another model that we trained, in this case not on three-dimensional images, but on two-dimensional max projection images. Large values of beta favor sparse representation, but lead to poor reconstruction, whereas smaller values of beta means a complex representation and often a good reconstruction. Here you see 5 actual cells for reference, and here we see generated cells. 3 of each for 7 different values of beta. And you see that at high values of beta, the cells begin to look alike, and they begin to look like more or less like a blurry object. This could be considered a blurry mean cell, if you will, in our model. Okay, another interesting experiment. So in a way, we predict this third channel, this GFP intensity of a subcellular structure from the two reference channels, right? The nucleus and the cell membrane dye reference channels. Depending on the subcellular structure that we are trying to predict, one can ask the question, is one of these two more informative than the other? Is the nucleus or the cell membrane more informative for the shape and location of the mitochondria? So here we did an experiment to remove the information of either the nuclear channel or the membrane channel using a permutation analysis, and see how the prediction changes. In these plots, the dots are once again cells. On the x-axis, we have a metric, which is the coupling to the cell. How well does the cell membrane help you predict your organelle of interest? And on the y-axis, we have the same thing, but then for the DNA or for the nucleus. In the top row, we are seeing a bunch of controls. What we did, instead of using actual GFP of organelles, we put in a blank channel, or we copied the actual DNA channel or the membrane channel. With a blank channel, we see that neither the cell nor the DNA show a very high coupling. When we have the DNA channel there, we see, of course, that there's a very high coupling to DNA or the nucleus, and vice versa with the membrane. Now, if we look at some actual organelles, we see that we have low coupling for the mitochondria for both the DNA and for the cell, which also is an indication that we are actually not doing a particularly good job of predicting the GFP intensity and by extension the shape and location of the mitochondria using these two channels. When we look at the nuclear envelope, which is around the nucleus, there, of course, we see, and this I think is a great, positive control, that there's a much stronger DNA coupling than coupling to the cell membrane, because most of the points are above the diagonal. Interestingly, when we look at the mitotic cells in blue, this relationship no longer holds, which also makes sense because the nuclear envelope actually breaks down during certain stages of mitosis. Finally, looking at the tight junctions, which are on or near the cell membrane, we see that the cell coupling is larger than the DNA coupling. We've done this, of course, for all the organelles that we have, which gives us a nice rank ordered list with lots of positive controls. But I want to emphasize that the ones which are in the middle, in many cases, we cannot simply predict them very well with a model that we have at this particular point in time. The tubular structures and the more punctated structures are difficult to predict. We have some ideas of how to improve that, but this is where we are at this point in time. Finally, and briefly, we ran we ran some out of sample data through the trained models. So specifically, we ran a small number of cell images of drug-treated cells through our model. Used two drugs, profeldin, which is a Golgi protein trafficking inhibitor, and paclitaxel, which is a chemo drug that stabilizes the microtubule and protects it from disassembly. We applied this to 3 different cell lines, microtubule tagged cell lines, Golgi and tight junction tagged cell lines. One important observation when looking at the latent space for the reference channels for the DNA and for the cell is that these drug-treated cells end up in the same part of the latent space, right? So this means that these cells have similar shapes in terms of their nuclei, in terms of their cell shape. Looking at target models, we do observe a shift in the conditional latent representation, which represents, in this particular case, the effect of paclitaxel on microtubules. By sampling images uniformly across this red arrow, moving from left to right here, we see the change from a uniform to a more condensed localization, which fits with the understood mechanism of action for this particular drug. Okay, future directions. So this was a project that grew over many years. And so we had a very, if you will, organic code base using a lot of old libraries and a lot of redundant stuff. We have been going through the effort of making this into a scalable and modular tool that can be incorporated into the automated and larger workflows that we have going at the Institute. Perhaps not the most fun work, but very important for the type of work that we want to do here. Current application, as I mentioned, is a shape space analysis of nuclei during colony formation, and that's being led by Ritwik Vassan and Guillermo Pires in my team at this point in time. If we predict the location of two organelles in the same cell with our model at this point, they may simply be on top of each other. Can we think about implementing conditional interrelationships between these organelles such that we can move to a more sensible model, a more sensible integrated cell? We can think of physical mutual exclusivity constraints or take into account known relationships, perhaps between organelles. We are doing a lot of segmentation in this institute as well, and we of course have information about these segmented structures, such as their volume. Can we incorporate that into the machine learning model in order to make things more specific? Besides having single tagged cell lines, we also have dual tagged cell lines. Here we see in yellow the nuclear laminar, and around that in white the ER. Can we use the dual and triple edited cell lines, either as part of the training or as part of the validation? And finally, we want to study the generalizability of these models. In a previous version of this symposium, I also very briefly presented this slide. This is a picture of a refrigerator. Then we paint, if you will, the refrigerator, the cell in magenta of the refrigerator and the nucleus, and then we run it through our generative model, and here you see the mitochondria of the refrigerator. This is of course absolute nonsense, but what is sensible in terms of extrapolating trained models either by themselves or using transfer learning approaches? So we have trained and can apply these models on the iPSCs. We have applied them to time-lapse colony formation movies of the same cells. We have shown that the model generalizes to some chemical perturbations and we can learn interesting things. Can we use the trained model for cardiomyocytes or perhaps external data? This is a third important and current research question that I wanted to bring up. In concluding this presentation, we want to acknowledge all the authors on this work, especially Greg and Rory, who ideated and implemented this approach and were really the main scientific driving force behind this project. We wish to thank Paul Allen for his vision, encouragement, and support. And finally, the team that is the Institute for Cell Science. This work was really only possible with the expertise and dedication of researchers from all the various disciplines that are combined at our institute. Thank you so much for your attention.
There's already a question here for you, Theo, so I'm going to read it out loud. So this is from Vasil, and he's asking if you are planning to use correlative light microscopy to make your models with higher resolution, so CLEM, basically.
Thank you so much for that question. I do not know what correlative light microscopy is.
I'm sorry, correlative light electron microscopy. Sorry, that was my bad.
Okay, well, so I've heard about electron microscopy, of course, going at a much higher resolution. What we have at this point, which is not electron microscopy, but is lattice light sheets. One thing that we are seeing, if we go to higher and higher resolution, is that our GPU cards simply cannot fit the images anymore. For these images, these three-dimensional images, it was already, quite a tour de force to get things to train on pretty big GPU cards, 32 gigabyte cards. So I think that one big challenge that we will all face as a community in one way or the other is that as we develop better and better microscopes and get, more high resolution images, is how are we going to apply deep learning to these, in the sense that it seems that although the field of deep learning and also the technology and hardware is improving very fast, I think that we are being outpaced by what microscopes can do at this particular point in time. So yes, we will continue to work with more high resolution microscopes. At this point in time, we don't have the idea that we would work with electron microscopes, because our institute really wants to stick at the level of organelles for the time being. I think that we have to come up with very creative solutions in order to deal with the more high resolution images that we are going to be confronted with in the coming years. It's my very general answer to that question.
All right, thanks, Theo. Everybody just keep adding in more questions as you see them. So I see another one here, Theo. I'm going to read this one out loud too. From Mary, I'm thinking of a recent preprint from your group describing integrated modeling based on spherical harmonics and PCA. Could you compare and contrast that method with the machine learning generative model that you displayed today? It seems like the goals are pretty similar. So are these approaches parallel or complementary?
Yeah, this is a great question. And you are absolutely right, Mary. And so these approaches are very complementary to each other. And that actually at this point in time, we are comparing them directly to each other under the umbrella of shape space analysis. So both with an approach where you first recognize the object by doing a segmentation, then mesh it, and then parameterize it. And then you can do all sorts of fun things with these shapes. We are trying a parallel approach where we are either trying to push the images as such through an autoencoder, which I think is a very strong way because it actually works. But also taking the approach where we start with parameterized objects ourselves and running them through the autoencoder model. What we did in that particular paper is we used principal component analysis.
We kind of lost you in the middle of your response. Did you want to do a really quick recap, or was that it?
Yeah, the really quick recap would be very great, sharp observation, Mary. Yes, we are pursuing various ways in which we want to describe the shapes of our cells, nuclei, and organelles. And we're doing that in a way in which you have read in this bioRxiv paper, using the spherical harmonic analysis and the PCA followed by that, as well as with the autoencoder. Of course, the hope with the autoencoder is, since it is so good at capturing variation and in a nonlinear manner, is that we may have fewer dimensions in which we can accurately describe and also biologically interpret the shapes of the organelles and nuclei in these projects. So we are actually directly comparing them, and it's quite exciting.
All right, thanks, Theo. Virginie, I think you had your hand up, so if you wanted to pop on the screen here and ask your question.
I'm also happy to send an email to Theo if we are short on time.
You can go ahead. We have a bit of time.
Okay, great. This was a really exciting talk, and it's super interesting work that you're doing. So thank you for that. I'm very much looking forward to read all about these different things you told us about. Just one question that I had on the spherical harmonic decomposition and PC analysis afterwards. So the thing with the so with the spherical harmonics, obviously, if you you have to set a cutoff at the number of coefficients that you take, right? And I mean, this is like the first coefficients are very much smoothing the actual objects. So can you comment on that? As in, what do you see is kind of the right number of coefficients? Do you think that, would this approach be amenable to more complex structure than nucleus, where there may be more high frequency information and where you may need to take more coefficients, or is that not really a problem? Sorry, that's a bit specific.
No, I think this is a great question. Thank you so much. No, you're absolutely correct. In a spherical harmonic decomposition, as most people, I guess, intuitively know with a Fourier decomposition or transformation, the first coefficients describe the lower frequency components with spherical harmonics applied to a nuclei or a cell. This means the rough shape, indeed, of the nucleus and of the cell. we know the resolution of our microscopes, and we know that there is a relationship between the coefficients and the actual sort of spatial resolution. So this is how far we were able to push it, right? And so we have a very accurate description at the level of the resolution of the microscope. For modeling purposes, one may actually tune it down a little bit and say perhaps we don't want to include these high frequency components and do it with more low frequency components. For other organelles, the spherical harmonic representation may not be the good one. And that's also why I was so excited about your talk, because there may be other representations which are much better. You know, if we think about tubular structures, or if we think about punctated organelles in the cell, then likely we would not go with spherical harmonics and be inspired by you and others to have other ways in which we model these.
This is super exciting in any case. Thank you very much.